Hello everyone, we're going to cover the fundamentals of genetics today. So first up to discuss is what even is genetics. So that is going to be the branch of biology that studies how traits are passed on from parents to offspring. Heredity is something that is very closely related to genetics because what heredity is, if you've ever heard of a disease being hereditary or something like that, what that means is heredity is the passing of traits from parents to offspring directly. So really, when somebody studies genetics, they are studying heredity. Okay, so let's talk about what a trait is. That is a genetically determined or inherited variant of a characteristic. So examples, looking at these two photos side by side, a trait might be hair color, eye color, skin color, shape of your eyes, nose shape, something like that. All of those things are traits with different variants in different populations. Next is Gregor Mendel. We have to talk about him. He's known as the father of genetics because he's one of the first people that really truly studied genetics and had some pretty important discoveries as well. So he's the pea plant guy, if you ever studied him in middle school. He's an Austrian monk in 1822, and his job at the monastery that he worked at was to be the gardener. And being a gardener at a monastery, he had a lot of time on his hands to look at plants. And so he noticed some things happening with the different pea plants that he was tending to, and he started doing experiments with those pea plants. So basically, there were different traits that the pea plants were expressing. They were different heights. The peas that they produced were different colors or different textures. So noticing that, he was noticing some traits popping up more than others, or potentially even two pea plants with certain traits having offspring pea plants that had different traits entirely. And so what he did was he took the flowers of the pea plants and he controlled their reproduction. So how plants reproduce is they have a male and a female part. So these anthers here that release the pollen that those are the male parts of the plant. And then this part in the middle, the pistil, that is going to contain the female parts of the plant. And so plants can fertilize themselves, or if they have an animal intermediary, they can fertilize other neighboring plants as well. So that's what bees do. They take the pollen and they spread it from flower to flower, and they provide that genetic variation. So what Mendel did was he chopped off those anthers and he controlled which plants were fertilizing which other plants. And so he did that because he was looking at inheritance patterns. If certain plants fertilized each other, what traits do their offspring have? And he figured out a couple of things. The law of segregation, which stated that a pair of factors are segregated during the formation of gametes. And so we talked about the law of segregation when we were talking about meiosis. And also the law of independent assortment, which states that factors for different characteristics separate independently or randomly of one another during gamete formation. So again, to summarize, those two things are closely related to one another. One just references the separation of homologous pairs and the other of the sister chromatids, but ultimately they both mean that there's random separation going on. So to remind you what a gene is, that is a segment of DNA on a chromosome that is going to control one trait. So you have a gene and it's going to code for one particular trait, not several. Allele is another term that you may have heard me reference before, but never really truly explain. And basically it's each of two or more alternate forms of a gene. So Mendel referred to them as factors because when Mendel was doing all this research, he didn't have all the information that we currently have about how genes and chromosomes and everything like that works. He was just looking at traits. So he called them factors, but he's actually talking about alleles. We use letters to represent those as well. So to give you a better idea of what I mean by the word allele, like let's use an eye color example. So me, I have brown eyes. That's what you can see looking at me. But I inherited a gene for eye color from my father, and I inherited a gene for eye color from my mother. And my father had blue eyes, and so since blue is a recessive trait, I know that he had to pass a blue-eyed allele to me. My mother had brown eyes, and I don't know exactly what her genotype was, but I do know that she had to pass on a brown-eyed allele to me because I myself have brown eyes. And if I know that I didn't get it from my father, it had to come from her. So I have one brown allele and one blue allele. And that's what I mean by that. They're different forms of a gene. Dominant and recessive are basically talking about the relative power of any particular gene. So some traits are stronger than others, and we call those dominant genes. And then other traits are weaker, and we call those recessive. So in the eye color example, we are talking about brown versus blue. Sorry, had to pause real quick and go answer a question of a student. Anyway, as I was saying, brown is a dominant allele as we're talking about eye color, and blue is a recessive allele when we are talking about eye color. So if you inherit both of those, a brown and a blue allele, your phenotype or your trait that's visible on your body will be brown. So me, I have brown eyes, but I know I carry the recessive blue gene, so I could theoretically pass that on to a child. But you would never know looking at me that I carry a blue-eyed trait. So if we are talking about representing that letter-wise, you would see for people who have two dominant traits, you would see two capital letters. If they have one of each, you'll see a capital and a lowercase. And if they only have recessive traits, you'll see two lowercase letters. Now, it's not always E's. It could be any particular letter, but this is how we identify the genotype or what the genes look like of any particular individual. So once again, genotypes are genetic makeups of different organisms. So represented by those letters that I was referencing in the slide before this. Phenotypes, on the other hand, are the organism's appearance. So let's say this fly over here, we were talking about what color its eyes are, and it has two green genes for eyes. So its genotype would be, let's say, homozygous recessive, but its trait that genotype controls, its phenotype is green eyes. Okay, so that's the difference between the two of those. With phenotype, though, it's not always something you can see necessarily on the surface of an organism. So you can have a phenotype of a particular blood type. So like I could have A positive blood, and that's my phenotype, but my genes that controlled that are something entirely different. So again, phenotype's not always visible, but it is a particular trait. To be homozygous for a particular trait, that means that you have two of the same genes. So either homozygous dominant is two capital letters, or homozygous recessive, which is two lowercase letters. And you can remember that because the prefix homo, that means same, and so you have two of the same things. Versus heterozygous, one of each, one capital, one lowercase, the prefix hetero indicating one of each, meaning different. So if you are homozygous, again, you have two the same, and if you are heterozygous, you have one of each. For a second, we have to talk about probability. because probability is the likelihood that any specific event will occur. So we use probability to predict what organisms' offspring will look like or what traits they'll have. So probability could be expressed as a decimal, a percentage, or a fraction, but ultimately it's the number of times a certain event might occur divided by the total number of events that might occur. So the way that we use probability to predict these things are Punnett squares. So you may have seen these in middle school or maybe not, but you'll have done something akin to this in math. Basically, we have two parents and we're going to cross their genotypes to figure out the potential genotypes of their offspring. So in this example over here on the left, the gray circles represent the parents, okay? So we have a homozygous recessive parent with our two lowercase p's, or little p, and we have a homozygous dominant parent with our big P over here. So what we do is we distribute those letters to the appropriate boxes. So anything that's up on the top is going to travel down, and anything that's over on the side is going to travel across. So this little p is going to go both here and here. This little p is going to go both here and here. And then this big P goes to this box and that box. This one goes to this box and that box. So that is a mess of arrows, but ultimately what you end up with in the middle is heterozygous offspring. Each one of these prospective genotypes has one of each letter, and that makes sense if you think about it, because this parent can only pass on little p's and this parent can only pass on big P's. So you have 100% chance of being heterozygous if those two organisms are the parent. Now with this example here, we have a homozygous dominant parent and a heterozygous parent. And so the chances are a little bit different with this cross. You have a 50-50 shot of being either homozygous dominant or heterozygous. This example here, we have a heterozygous parent and a homozygous recessive parent. And in this cross, we're seeing a difference in phenotype. Half of the offspring, half of the potential offspring are purple here, and half of the potential offspring are white. And what that means is you have a 50-50 chance of being heterozygous or homozygous recessive as a result of this cross. And then finally, we have two heterozygous parents, which produces the most variation of genotype. They could have offspring that are homozygous dominant, heterozygous, or homozygous recessive. And so you'll notice, though, that the recessive trait, the white flower, only shows up in homozygous recessive individuals. And so these Punnett squares are known as monohybrid crosses, meaning that you're only figuring out one characteristic, one gene, one trait is being tracked. And there are bigger crosses, dihybrid crosses and trihybrid crosses and so on and so forth, that you can use to determine more genes at the same time, but they get exponentially more complex as you go. Complete dominance and recessiveness is one form of inheritance where one allele, one form of a gene, is completely dominant over the other, meaning that in heterozygous individuals, when you have this type of genotype, your trait will match the dominant characteristic 100%. So an example of this would be tongue rolling. So some of you can probably roll your tongue into that little taco shape, and some of you can't. And I know you're testing that right now, probably. But this is an example of complete dominance, because either you can or you can't. You can't do it halfway. You can't kind of, sort of do it. You either can or you can't. You either have the necessary musculature or you don't. And then tall versus short pea plants. The pea plants are either tall or they're short. There's no mid-range there. So let's talk about this sample problem. we have a heterozygous purple flower with another heterozygous purple flower. So we are going to use that capital P is dominant, and that is going to be for purple. And then our little p is going to be recessive, and that's going to be for white. Okay, so if we have two heterozygous parents, both of our parents have one of each letter. So we have a big P and a little p, and then we have a big P and a little p. Okay, so we move everything on the top downwards and everything on the side rightwards. So this big P is going to move down to this box and this box because this P is going to go in that direction. This P is also going to move down. So we have a little p and a little p there. Okay. Then over here, everything over there is going to move in that direction. So this big P is going to go here and here, and this little p is going to go here and here. Okay. So what you're seeing is, again, potential offspring. It doesn't mean that if these pea plants had 4 offspring, they'd look exactly like this with this distribution. It's just percent chances. So what we are seeing is a homozygous dominant. We're seeing 2 heterozygous options and 1 homozygous recessive. So 1 out of 4 of those squares is going to be homozygous dominant. And 1 out of 4 written as a decimal is 0.25 or 25%. Okay? That's what that means. We see 2 out of 4 of these popping up as heterozygous. I know that this one is little p, big P, and that one's big P, little p, but ultimately it doesn't matter. So 2 out of 4 of them are heterozygous. And that's 50%, like this. And then finally, we have 1 out of 4 as homozygous recessive. And that's going to be 25% once again, little p, little p. Now, those are the genotype percentages. If we wanted to do phenotype percentages, well, we'd have to look at what each thing means. Since purple is dominant, if you have a dominant allele, you're going to pop up as purple. So this one has purple, this one has purple, and this one has purple. So 3 out of 4, or 75% chance of that offspring having the purple flowers. Whereas only 1 out of 4, or 25%, are white. So any particular offspring has a 25% chance of being white and a 75% chance of being purple. So that's all the information that you can tell with a Punnett square. Okay. Sample problem number 2, on the other hand, is a homozygous person with freckles and a heterozygous person with freckles. Okay. So what that means, let's use F's this time. So a capital F is going to be dominant and represent freckles, and a lowercase f is going to be recessive and represent no freckles. Okay? So if we have a homozygous person with freckles, they're going to be homozygous dominant and have 2 capital F's. And if you're heterozygous, you have a capital F and a lowercase f. And yes, I'm purposely drawing my lowercase f very curly so that you can see the difference. Okay? So quickly doing this cross, we distribute our capital F's downward, and we can distribute that capital F across and that lowercase f across. So here's what we are seeing. We are seeing 50% or 2 out of 4 as capital F, and that's going to be homozygous dominant. And we're seeing 2 out of 4 or 50% heterozygous, okay? Now, we know that the capital F, the freckles, is dominant. So every single one of these offspring are going to have freckles. So you have a 100% chance of having freckles if you have 2 parents that both have freckles, because that is a dominant trait, and every single possibility in this Punnett square has capital F's. Okay, so you have a 100% chance of freckles. Next thing I want to talk about is incomplete dominance. So we talked about complete dominance where one trait wipes out another, but that's not always what happens. Sometimes you get a blending of the two. So let's say, for example, we have a dominant trait and a recessive trait. Our dominant trait is red, our recessive is white. So that's what you're seeing here, okay? But in heterozygous individuals, instead of the heterozygous just being red because red's the dominant trait, instead you're seeing a blending of the two. And that's what we refer to as incomplete dominance. Because the red is not completely wiping out the white, it's blending instead. So you'll see this in flowers, you'll see it all over the place. But basically, what I want you to take away here is that in heterozygous individuals, instead of one trait being completely dominant over the other, they're not really dominant over one another at all, and instead they just blend together. Okay, so here's a flower example that you can see in the Punnett square associated with it. Another thing that you'll see in nature is called codominance, which is similar in the sense that heterozygous individuals are going to express both traits, but instead of blending together, they get expressed side by side. So in this 2 chicken rooster example, we have a black one and a white one. And if this were incomplete dominance, you'd get a gray rooster as the offspring. But instead, you get a black and white speckled rooster. So both of those traits are being expressed side by side rather than blending together. But again, you're only going to see that in heterozygous individuals. So you can see that in animals, in plants. You'll see it in flowers a ton because there's a lot of speckled flowers around. And then another form of inheritance that I want to just briefly touch on is called polygenic inheritance. And what that is when more than one gene controls a single trait. So you see that in honestly most of the traits in your body. So skin color, hair color, eye color, all of those are controlled by a lot of different traits. And that's not to say that every trait in your body has more than one trait that controls it, but a lot of what you can see on your body's surface is controlled by multiple traits. Like I think skin color is controlled by over 100 different genes. So whenever you are trying to figure out like where certain things came from, like if you have a certain skin color and you want to know where you got it from, you have to consider that there's hundreds of genes that are controlling that, or your hair color is a bunch. I think your hair color is somewhere around 50. Don't quote me on that. I'd have to look up the exact number. But again, a lot of different genes that are associated there. Another thing that I'd like to touch on briefly is called a dihybrid cross, and you'll watch an Edpuzzle about this, but a dihybrid cross is when you look at 2 genes at the same time. So let's say we're looking at fur color and length. So we have a parent that is heterozygous for both. Let's say capital S is going to be short fur, and then little s is going to be long fur. And then we have capital B for black fur, and then lowercase b for brown. So if we have a heterozygous parent, their genotype would look like this. And if we crossed that with another heterozygous parent, the other parent's genotype would look like this. So to set that up, we would have to draw a much, much bigger Punnett square. We'd have to draw it, I'm gonna draw it up here, but we'd have to put 4 spots. So our Punnett square is gonna look like that, and it's a dihybrid cross now. So to fill in the parental genotypes, you could have any number of combinations. So you could have a capital S, capital B getting passed on. You could have this as an option. You could have this as an option, or you could have this as an option. And that's the same for down here as well. So as I said previously, the amount of possibilities exponentially increases as you go. And so you would distribute these accordingly. So instead of 4 different possibilities, you could have 16. And then in a trihybrid cross, you might have 32 or 64 or whatever, and so on and so forth. So these, and again, you'll watch an Edpuzzle on this, but these are not harder, just longer, let's say. Anyway, I hope this was helpful. If you have questions, please ask me in class.